Based on that silence, it sounds like it's a good time to start. So I'd like to welcome you to the first of what hopefully will be many in the series of lectures for precision medicine. And so this will predate our founding of the Center for Precision Medicine. I'm very happy to have Bill Sellers as our first speaker for this series. He's a core member of the Broad Institute and affiliated with Dana-Farber, as well as Brigham and Women's Hospital. Bill trained starting at university at Georgetown and then at the University of Massachusetts, where he did his MD, training clinically at UCSF, and then going to Dana-Farber, where he worked as a clinical fellow in oncology, working initially in David Livingston's lab, and then with Bill Kalin, obviously two outstanding mentors in the area of cancer biology. And I'm not going to be able to go through all of the accomplishments that Bill has in the area of cancer. He's really played important roles in the area of basic cancer biology all the way through clinical work. And just make a few comments. He's asked to be on numerous advisory boards, including the NCI advisory board appointed by Barack Obama, and also advising on major cancer centers in the United States like St. Jude's Children's, as well as Damon Runyon Cancer Research Foundation, which is a wonderful organization, and Memorial Sloan Kettering, just to name a few. He has done a lot of work that you'll hear, the more recent work, so I'm not going to introduce that, but I've just historically would mention that Bill played an important role in identifying and understanding the role of PTEN biology in cancer. And when I joined in Boston, that was one of the areas he was quite active in. It was very exciting to see firsthand how the development of discovery for EGFR mutations with Matt Meyerson and others at Dana-Farber, and how that observation has now turned into obviously a major clinical area of therapy, and also understanding resistance in the area of non-small cell lung cancer. He also was involved in identifying MITF. I don't know if you remember all the accomplishments you have, but that was very exciting to see firsthand. And one thing I just wanted to highlight, you can't highlight everything, but one of the things that really impressed us when we were there is the joining of technologies with the understanding of biology. And so as an example, a development of SNP arrays that were used for genotyping, Bill's lab played an important role with Matt Meyerson and others in using it to look for copy number alteration in cancer. Today we use it without thinking about it. I mean, we have various ways of looking at copy number alterations, but really at the time that was cutting edge and really developing and working with industry also to develop and work on the next chips, as well as some of the first examples of developing the computational biology algorithms to help analyze the data. Since when he moved to Novartis as a vice president for oncology, global oncology, he's had a very accomplished tenure there before coming back to academia, where he was involved in the development of 40 therapeutic drugs, including the CAR-T program that was done in association with the University of Pennsylvania with Carl June, a development of a PI3 kinase alpha inhibitor, as well as SHP2 and BCR-ABL allosteric inhibitors, just to give some examples. And so today we're going to hear about some of the things Bill's currently doing. It's really exciting to have him here, and I appreciate it very much. Thanks, Bill.
Hello? Yep. Okay. So I'm mainly going to talk about work that I was doing at Novartis, plus some new data around a project that we've called the Cancer Cell Line Encyclopedia, which probably people have heard of. So over the last few years, both at Novartis and now at my new group, I've been interested in a number of questions relevant to therapeutic development. And I'll try to illustrate some of the approaches both to target discovery, asking the question what genes are necessary for the maintenance of the cancer phenotype. In drug discovery, how do we discover new ways to drug validated targets? I think people know that as we've discovered many new cancer-dependent genes, not all of them are as easy to drug as a kinase, so how do we think about that? And then therapeutic resistance, how can we discover combinations that have not just anti-cancer responses, but really have curative potential, and what will be the elements that are important for thinking about that. So one of the constructs that I've been interested in is this cell line encyclopedia. So when I moved to Novartis, most drug discovery programs that were running in industry, including those that had led to the discovery of EGFR inhibitors, were really using just one or two cell lines. So all the EGFR inhibitor drug discovery efforts that were ongoing at AstraZeneca and other places really relied on one cell line, A549, which turned out not to be EGFR mutant. And therefore, at the time that EGFR inhibitors went into the clinic, no one understood that there might be a subclass of cancer that was particularly susceptible to EGFR therapeutics. So when I arrived at Novartis, I established a collaboration with basically my former group at the Broad Institute to collect 1,000 cancer cell lines, mostly from commercial sources, and annotate them by genetic expression data sets. So over time, we've been using that cell line encyclopedia to query those genes that are now required for the ongoing growth of those cell lines using large-scale shRNA experiments. And the Novartis experience in this was published last year in Cell, outlining a project that we called Project Drive which stood for the deep RNAi interrogation of viability effects in cancer. And essentially what we did was to synthesize oligonucleotides encoding the shRNAs targeting roughly 7,800 genes. These were cloned into viral pools and then used into plasmid pools and used to generate lentiviruses that were then infected into cell lines. And after 5 doublings, we would count the abundance of each shRNA by next-gen sequencing. Project Drive was the culmination of a lot of technical development in the end, we targeted 7,837 genes with an average of 20 shRNAs per gene. And we took that library and profiled the viability effects in 390 cell lines. So I think people know in the early days of shRNA screens, a lot of false positives were found because of the off-target effects of shRNA, and I hope to convince you, and I hope if you've looked at the data sets, that when you use 20 shRNAs per gene to understand the effect of each depletion of each gene, the data are far more robust than when you're using smaller libraries. So if you have 20 shRNAs per gene, you have to decide how you're going to call the effect of each gene's depletion on the viability of each cell line. And we used an algorithm called ATARIS that was developed at the Broad to take, sorry, I'm trying to find the pointer here, to take the individual shRNA profiles for a roughly average of 20 shRNAs per gene and coalesce those into a single gene score. So from those 20 shRNAs, you would take the consensus pattern across 390 cell lines and develop a single ATARIS score, dropping out shRNAs that had patterns that were incoherent with the overall voting pattern. So you could imagine we would have 17 shRNAs that looked similar, 3 shRNAs that looked divergent, and the 17 would become the consensus pattern for each gene. And you can see what it looks like in looking at the data set for KRAS. So these are all the individual shRNAs for KRAS. You can see here there are some shRNAs that have divergence, but in general there's a very noisy pattern. But when you take all 20 shRNAs, convert them to the Atara score, and then rank plot the Atara score for each cell line, and then color each cell line by whether or not they're KRAS mutant, you can see with a p-value of 10 to the minus 16th, the number one predictor for dependence on KRAS by shRNA is in fact KRAS mutation. This doesn't seem particularly insightful, but at the time, there was actually a lot of debate about whether or not KRAS dependence, as measured by shRNA, was in fact strongly correlated with KRAS mutation or not. And that's because a lot of the early screens, again, were sort of underpowered. So in Project Drive, in the publication in Cell, we outlined by Atara score those genes that had a strong outlier phenotype, and we roughly categorized those into 4 categories. Those that were genetic drivers, those that were expression-based drivers, where the top biomarker would be expression of the given gene, metabolic outliers, and then synthetic lethal outliers. And for the purposes of today's discussion, I'm not going to talk about the genetic drivers or the expression-based drivers. I'm really going to focus on metabolism and then synthetic lethality. So I think metabolism is a particularly tricky area to work in with respect to cell culture because, of course, you're in an artificial environment. But recently, we've finished the metabolite profiling of roughly 930 of the cancer cell lines that are in the encyclopedia. And so we're now able to provide the metabolite level data and how it intersects with cancer dependence. And I want to focus on one of those genes, asparagine synthase, as an example of actually some of the problems around doing metabolism data in cell culture. So as I mentioned, one of the new data sets we now have for the cell line encyclopedia going on beyond genetics and expression is a global metabolite profiling of 225 polar metabolites profiled in 928 cancer cell lines. And the manuscript preparation and the analysis is being led by a very talented graduate student named Hao-Shin Lee, and all the metabolite profiling was done at the Broad Metabolomics Corps run by Clary Klisch. So this is a hierarchical clustering of all 920 cell lines by metabolites. And so I think the first thing you can see is there's a strong dichotomous breakdown of cell lines into metabolite space. And if you look along the bottom, you can see that this is the color of the lineage of all these cell lines, and that there's a strong lineage distinction in this metabolite space. And this turns out to be mostly hematologic cell lines. Now, the problem with that is most hematologic cell lines are growing in suspension. So we're not sure if this large bifurcation is either hematologic or is it suspension cell lines. But for the purposes of the subsequent analyses, we treated these two groups independently so there wouldn't be confusion about certain metabolites scoring in some comparisons just because they were either heme or growing in suspension. So we have that one issue, which is suspension cells and or heme cells have a very different profile than adherent cells. The second issue is that adherent cells often grow in different medias, and we chose to use the medias that were recommended by the providers of all the cell lines. Partly because all the expression data we had already generated was in the recommended media, and all the functional shRNA and CRISPR data were also done in the recommended medias. So we felt like using the recommended medias for the metabolite profiles was also critical to having an integrated data set. But that also meant we were aware that the differences in media might contribute to differences in the metabolite profiles. And here you can see a ranking of metabolites that are influenced significantly by what media the cells are growing in. So this is a rank order of all metabolites, those 225, ranked by whether there was or wasn't a media effect. And you can see certain metabolites like hydroxyproline, threonine, valine, phenylalanine are significantly elevated in the intracellular compartment only due to what media they happen to be growing in, or not only, but mainly due to what media they're growing in. And for example, I pointed out asparagine synthase, and asparagine turns out to be highly media dependent when you measure the metabolite concentration of asparagine. We're then able to look at the CRISPR data now from the Broad Institute and ask whether or not there are dependencies in cancer space that are also media dependent. And here you can see a ranking of genes whose essentiality or viability by CRISPR knockout is highly dependent on what media they're growing in. And I'll point out here asparagine synthase, which was a hit in the shRNA Project Drive, is also a hit in CRISPR space, but it's highly media dependent. And that's, of course, consistent with the idea that asparagine itself is highly media dependent. And so if we look at this now in more detail, you can see that the top correlated metabolite that strongly is associated with the knockout phenotype for asparagine synthase is in fact asparagine. If you look at the intracellular concentrations of asparagine, there's a more than log difference over the various cell lines that is strongly correlated with the media concentration of asparagine. So for example, here at 0.38 millimolar is the concentration of asparagine in rPMI, and here at 0 is the asparagine concentration in DMEM. So DMEM turns out to have no asparagine, rPMI has 380 micromolar asparagine, and then that has a more than 1 log difference in the measured metabolite concentration intracellularly. If you then look at whether the intracellular concentration of asparagine is or is not correlated with sensitivity to asparagine synthase knockout, you see a strong correlation between the intracellular asparagine concentration and its dependence on asparagine synthase, meaning that if you're a cell line like A2058 growing in DMEM with no asparagine, you become hyper-dependent on asparagine synthase to maintain the levels of asparagine. And you can see that here when we do it by shRNA knockdown in A2058, we can take 2 different shRNAs, knock down asparagine synthase, and these cells become completely dependent on asparagine synthase. If we now add back non-essential amino acids, which include asparagine, we can completely rescue the viability effect of knocking out asparagine synthase by adding back asparagine. But if we now deplete asparagine from the non-essential amino acids by asparaginase, now the viability effect is restored. So that's a long way of saying that the effect of a depleting asparagine synthase in CRISPR space is simply an artifact of the media that we're growing the cells in. And I'll just point out that a lot of things that people study, like calcium and magnesium transport, shown here, the ferritin transporter, zinc transporter, bicarbonate, are all media effects in cell lines. So I think this just underscores how important it is to validate various metabolite dependencies in an in vivo setting. So if we now eliminate the confounding effects of media and we take out those dependencies which are media effects, we can ask, are there specific metabolite alterations that are now correlated with genetic effects independent of the media? And you can see a number of strong outliers where a mutational event is associated with a strong metabolic signature. And there are 2 here that I'll point out as examples, but there are several others which I won't go over. The first is, of course, IDH mutation. We know that IDH mutation is a mutant which creates the gain of function ability of cells to generate 2-hydroxyglutarate. And in fact, if you look at 2-hydroxyglutarate, it is a strong outlier strongly associated with IDH mutation, and all 5 cell lines that are high for 2-hydroxyproline are all IDH mutant. So this is discovered or rediscovered in this data set. It's a known feature. It just says that it's also robust in this data set. Similarly, we know that KEAP1 is associated with glutathione alterations, and you can see here KEAP1 mutation is strongly associated with higher GSH levels compared to KEAP1 wild-type cells. So these are 2 very strong genetic effects that lead to certain metabolic signatures. As I said, there are others which I will not cover here. One of the strongest correlations between metabolism and alterations at the genetic level and or the methylation level is the finding that specific methylation events in different amino acid transporters are strongly correlated with metabolite outliers. So for example, here you can see the expression of the carnitine acyl carnitine translocase. Here's the RNA level of that transporter. It's high in cells that have no methylation, and it's very low in a small group of cell lines that have strong methylation of the promoter of this gene. And if you now look at the hierarchical clustering, what you see is a cluster of acylcarnitine alterations which are strongly altered in the setting of methylation of the translocase. So this particular translocase is responsible for translocating acylcarnitine across the mitochondrial membrane, and when that promoter is methylated, this builds up in the cytoplasm and gives this specific metabolite signature shown here. There are a number of other methylation events that also cause very specific metabolite dysregulation in the data set, and you'll be able to take a look at those online shortly. So the other interesting area in the metabolism space is whether or not specific oncogenic driver mutations or tumor suppressor mutations are or are not associated with specific metabolic dysregulations. And there's been a lot of published literature around, for example, KRAS being associated with certain metabolic signatures. So we looked specifically at the known canonical mutations and were unable to really find strong metabolic outliers associated with specific mutations. So for example, you can see EGFR mutations, essentially no distinct metabolic signature, KRAS mutations, no metabolic signature that passed a threshold, same with NRAS, same with tuberous sclerosis or PTEN, and P53 had a number of metabolites that crossed the threshold, but we don't really think this is statistically meaningful, even though it's barely above the threshold. So this is quite interesting. It either means that there really are no defining metabolic outlier profiles that uniquely identify certain mutational events, or it means that they all have a similar metabolic profile and we don't have a comparator to allow us to distinguish the fact that all of these might share a metabolic profile compared to, for example, a normal cell. So we didn't have a normal comparison. But I would say in this data set, it's not clear to us that there are strong oncogene-specific metabolic profiles. There might be a general profile that would be indicative of oncogene addiction, but not one that's specific to each mutation. Okay, so in this area of metabolic dependencies, obviously it's very important that hits like asparagine synthase are validated in vivo, and I would say that's true for all of these other hits. So for those that are working in this space, it's really critical that one then go on to do in vivo validation. The area that I want to spend a little more time on is synthetic lethality, because I think there's also a lot of interest in that area with respect to, for example, the synthetic lethal interaction between BRCA mutation and PARP inhibitor sensitivity, and we were interested in whether or not new synthetic lethal interactions could be discovered. One of the most common synthetic lethals we now see both in the RNA data sets and the emerging CRISPR data sets are a class that we would call paralog dependence or paralog synthetic lethality. And this is the case in which there are two essential genes, for example, the essential paralogs ARID1A and ARID1B, in cell lines that turn out to have a mutation in ARID1A, shown in red, they become dependent on the remaining paralog ARID1B. Similarly, for the paralog pair RPL22L1 and RPL22, mutations occur in mismatch repair mutant colon cancer in RPL22 because it has an MSI locus, and those cell lines become dependent on the paralog pair RPL22L1. Otherwise, this would be an essential gene for all cells if you knocked out the pair, but because one of the two paralogs is already mutant in a cell line, they become especially dependent on the remaining paralog. And the same is true for R4 and 5. So the most interesting one we've discovered so far is a dependence, a synthetic lethal dependence on PRMT5, and I'll spend the next 10 minutes or so discussing that specific synthetic lethal interaction. So a number of people have understood that co-deletion of genes might lead to particular vulnerabilities in cancer. And one of the ones that's drawn interest over time is the fact that when CDKN2A or P16 is deleted, you often co-delete the gene methylthioadenosine phosphorylase. And this is of interest because these two genes are separated only by 100 kb in genomic space, which means most of the time that you delete P16, CDKN2A, you also delete this metabolic enzyme methylthioadenosine phosphorylase. And you can see that illustrated here in glioblastoma. CDKN2A is deleted in about 60% of GBM, and 50% of the GBMs also co-delete MTAP. So about 80% of the time you delete P16, you'll also delete the MTAP gene. Now, what's MTAP? It's in the methionine salvage pathway, and it's responsible for converting methylthioadenosine to MT1RP. It basically leaves the ribose and cleaves off the nucleotide, as you can see in this reaction. That's a critical step in the methionine salvage pathway, and the methionine salvage pathway is critical for the generation of SAM, which is the cofactor for methyl donors reactions driven by methyltransferases, such as PRMT5. So one possibility is if you delete methylthioadenosine phosphorylase along with P16, you would somehow become hyper-dependent on other enzymes in the methionine salvage pathway. So we took a directed look at that data by dividing cell lines into those cell lines that had deleted P16 and MTAP versus those cell lines that had not, and asked whether there were specific dependencies associated with the deletion of MTAP. And the first one that emerges is MAT2A, which is in fact in the methionine salvage pathway. So cells that delete MTAP become hyperdependent on MAT2A, and this is the basis for a therapeutics program at Agios Therapeutics, who also made this discovery contemporaneously, and they're now in the clinic with a MAT2A inhibitor to treat MTAP-deleted tumors. However, what you can see is that wasn't the top scoring gene. In fact, the top scoring gene was PRMT5. We didn't know why this was, but what was interesting is in the literature, PRMT5 always coexists with its heterodimeric partner WDR77, and WDR77 was also a hit in this screen. That heterodimeric complex actually exists as a heterooctamer of PRMT5 and WDR77, then associates with adapter molecules that allow it to recognize substrates, and those adapter molecules can either be Rio kinase, which allows PRMT5 to then methylate nucleolin, and you can see that Rio kinase was also a hit in this screen. PRMT5-WDR77 can also associate with a member of the spliceosome called PICLN, allowing PRMT5 to methylate the spliceosome, and PICLN was also a hit in this pathway. So not only did we get PRMT5, we got its obligate heterodimer WDR77 and 2 of the substrate adapter proteins. There's a third substrate adapter, which is known to be an important regulator, mediator of PRMT5 methylation, which was not in the shRNA screen. So we first tried to validate the dependence on PRMT5 by using DOX-inducible shRNAs in a non-pooled setting. So you can see here we took a MTAP-deleted pancreas cell known as MIA-PACA2 and depleted PRMT5 by shRNAs, and you can see a knockdown effect that impairs viability. If we then rescue that effect with a cDNA that has a mutation, so it's now resistant to the effect of the shRNA, we completely rescue the effect, suggesting this is not an off-target effect of the shRNAs. In addition, because these cell lines have 2 events, they have a deletion of P16 and a deletion of MTAP, in order to ask, was it specifically the deletion of MTAP that conferred susceptibility, we made an isogenic pair in which we knocked out MTAP by CRISPR knockout, leaving P16 intact, and again showed that that became sensitive to PRMT5 depletion. I'm not going to show you that data. We then went on to show that in vivo, if we take an shRNA to PRMT5 in a MTAP-deleted cell, we can get regression, shown here, as compared to the non-targeting control. But we also showed that in an MTAP wild-type cell, the shRNA to PRMT5 did not have an effect on viability. So in vivo, we have a synthetic lethal effect on loss of viability in the MTAP-deleted setting. So the question is, how does that work? So we have deletion of MTAP, some perturbation of the methionine salvage pathway, and then some dependence on PRMT5, but we don't have a biochemical connection between the MTAP deletion and some excessive dependence on PRMT5. So the first question is, when you delete MTAP, the prediction is the substrate, MTA, should accumulate in cells. That's our first prediction. And you can see here, measured by mass spec, intracellular levels of MTA are substantially higher in the MTAP deleted setting versus the MTAP wild-type setting, and this includes the isogenic pair where we knocked out MTAP and you show a substantial increase in the amount of the substrate known as methylthioadenosine, or MTA. And I showed you previously that MTA and SAM are connected in the methionine salvage pathway, and MTA, in fact, is structurally very similar to SAM, just lacking the carboxylic acid and the amino group, but retaining the sulfhydryl group and the methyl group, shown on the left side of methylthioadenosine. And in fact, people had shown previously that MTA could act as an inhibitor of the PRMT5 family. So our hypothesis was that MTA was building up in cells and acting as a PRMT inhibitor, but still it hadn't been shown that this was specific to PRMT5. So we took MTA and asked in vitro, is MTA an inhibitor of methyltransferase function? And you can see in the top panel, MTA is a selective inhibitor of PRMT5 while sparing other PRMT family members as well as a broad range of other methyltransferases. And that's compared to sinafungin, which is an analog of SAM that acts as a broad methyltransferase inhibitor. So somehow MTA selectively inhibits PRMT5. And to us, this was a big hint as to why PRMT5 would be specifically required in the MTAP deleted setting. But we still didn't quite understand how that would work structurally. So we solved the crystal structure of MTA bound to PRMT5, and I'll first show the crystal structure here on the left of sinafungin, the SAM analog bound in the catalytic site of PRMT5. So the important thing to look at is this glutamic acid E435 here, which is coordinating the substrate arginine shown here. So this is the arginine of the substrate that will get methylated by PRMT5, and the methyl donor group would be positioned right here. So this is the poised transition state of where the methyltransferase would be added to the glutamic acid. And you can see at the bottom of the pocket, the carboxylic acid of SAM is being coordinated by a lysine and a tyrosine. However, in the MTA-bound state, there's no carboxylic acid coordinating those two residues, and instead, the glutamic acid that would normally coordinate the substrate arginine is deflected downward and is now coordinated by this lysine and tyrosine. And we believe that this is the MTA-inhibited state of pure MT5. So still, how does that explain MTA as a selective inhibitor of PRMT5? It turns out those two residues at the bottom of the pocket, this lysine and tyrosine that I showed you, are coordinating this glutamic acid in the MTA structure, while they're conserved between human and Drosophila PRMT5. They're not conserved in any other PRMT family member. So we believe that this MTA-inhibited structure is unique to PRMT5 among the other PRMT methyltransferases, and explains why in cells that delete MTAP and have high levels of MTA, PRMT5 might also be partially inhibited. So I'm going to skip that in the interest of time and go to the model, which is that in wild-type cells that have a normal MTAP gene, this would be the function of PRMT5. It would be coordinating SAM and transferring methyl groups to its substrates. In the deleted setting, you get an increase in MTA, which I've shown you can occupy the PRMT5 pocket and act as a partial inhibitor of PRMT5, lowering the functional activity of PRMT5 in those MTAP deleted cells. If you now come in with an shRNA, the shRNA further depletes the functionality of PRMT5, but in the wild-type setting, there's sufficient PRMT5 activity to not affect the viability of those cells, whereas in the MTAP-deleted cells, since PRMT5 in our model is already partially inhibited, the shRNA depletes PRMT5 and you lose viability. The prediction is if you knock out by CRISPR PRMT5, basically all cells would be dead because this is an essential enzyme. And if you now look in CRISPR space, in fact, one of the big differences is in CRISPR space, PRMT5 shows no selectivity to MTAP-deleted cells versus an shRNA. So it's an essential enzyme, but partial depletion by shRNA reveals this synthetic lethal phenotype. We're encouraged by this because most inhibitors that are developed therapeutically are not full inhibitors of an enzyme. So we think that this leads us to believe we can create an inhibitor that would be targeting this MTAP-deleted setting. So that's one of the more interesting synthetic lethals we have discovered, and both at Novartis and in my new lab, we're trying to think about how to exploit that from a now therapeutic point of view, not just from a genetic point of view. So the second question I mentioned is, from a drug discovery point of view, there are lots of validated therapeutic targets now, for example, MYC and RAS, and one of the real issues is how do you take those on from a therapeutic point of view? And I just want to talk about briefly our program targeting the SHP2 oncogenic phosphatase as an example of how some clever biochemistry really helped to drive a discovery of a novel inhibitor that was really works by an unanticipated mechanism. So SHP2 is one of the rare phosphatases which is not a tumor suppressor. Instead, it's an oncogene. Under most circumstances, SHP2 is in an inactive state, but if you have mutations in SHP2, which have been found in juvenile myelomonocytic leukemia and occasionally in breast cancer and other solid tumors, these mutations activate SHP2 by disrupting the inactive conformation of SHP2. So there was already data suggesting that SHP2 might be a driver of cancer through oncogenic activating mutations. Normally, when it's not being mutated, SHP2 plays an important role in coupling the activation of receptor tyrosine kinases to the activation of the MAP kinase pathway. So, for example, when a receptor tyrosine kinase like FLT3 or others becomes active and phosphorylated, it recruits SHP2 through its C-terminal and N-terminal SH2 domain, activating SHP2 and triggering activation of the MAP kinase pathway. So in RTK signaling, SHP2 is also thought to be a critical and important mediator of both normal RTK signaling and oncogenic RTK signaling. And this importance in the RTK signaling has been highlighted by a number of studies showing that RTKs can provide adaptive resistant mechanisms to therapeutics. So for example, Todd Golub showed if you treat melanoma cells with a BRAF inhibitor and then look for secreted ligands that can rescue cell viability and create resistance, HGF was a top hit. So HGF and the MET receptor rescued BRAF inhibitors in melanoma. At Novartis, we did several screens using secretomics approaches and found that if you had a MET inhibitor, FGF and neuregulin could rescue cell viability and create resistance. Conversely, if you had a FGFR inhibitor, now HGF and neuregulin and EGF could rescue, suggesting the possibility that when you inhibit an RTK, there will be bypass activation of receptor tyrosine kinases that alleviate the initial inhibitory activity. And finally, René Bernard showed that in the setting of BRAF inhibitors in colon cancer, in fact, SHP2 was an important mediator of this RTK bypass resistance mechanisms. So these three studies suggested to us the possibility that in addition to its normal function as a driver of RTK signaling to the MAP kinase pathway, SHP2 might be an important mediator of adaptive resistance to RTK inhibitors. And therefore, it might be an inhibitor of SHP2 might be a combination agent to use in combination with RTK inhibitors like EGFR or FLT3 inhibitors. In the DRIVE data set, which I mentioned earlier, this association between RTK and SHP2 dependence was also borne out in that if we looked at those cell lines that had mutations in RTKs, either amplification, mutation, or translocation of various RTKs, there was a strong association with SHP2 dependence and RTK-driven tumors, whereas the reverse was true for RAS mutant tumors. So those led us to the idea that we wanted to inhibit SHP2 from a therapeutic point of view, and I'd say we did what 90% of pharmaceutical companies would do, is we first made an assay where we truncated everything but the phosphatase domain of SHP2, got rid of all the inhibitory signals, and made a superactive SHP2 protein, and then used that in an HTS screen, and we got no tractable hits. And this is because, as many people know, there's a very electrophilic cysteine in the catalytic site of phosphatase, and you get tons of false positives when you screen for pure catalytic inhibitors for phosphatases. So in parallel to that effort, we also made a second assay, which really was the brainchild of Pascal Fortin and his biochemistry group, where it took time, but we were able ultimately to produce purified full-length SHP2 and then incubate it with a synthetic peptide containing 2 phosphorylation sites and show that that peptide was capable of activating the otherwise apo SHP2 into the active form of SHP2. And so we then took that screen, that protein and assay into a second, in this case, pilot screen, which I'll show you here, and screened 100,000 compounds looking for inhibitors of the second assay. Now, one thing I've seen in returning to academia is lots of people doing HTS screens, and then it seems to me miraculous that they can then pick out a hit and start working on it. Because our experience with HTS screens are more like this. So this is a screen of 100,000 compounds. This is actually not the HTS. This is the pilot screen before the HTS. HTS in companies is usually 1 to 2 million compounds. So the 100,000 screen is a test of the assay and its reproducibility. And you can see at a certain threshold of inhibitory activity, there were roughly something like 5,000 hits, of which 6 ultimately validated from that first hit list of 5,000 compounds. One of them, I'll show you, SHP-836, is illustrated here in the confirmatory assays, which were a full dose response curve by IC50. SHP-836 inhibited the peptide stimulated activity of the full-length protein, but had no effect on the catalytic domain only. So we had this catalytic domain, then we had the full length, had no activity on the catalytic domain only, but had inhibitory activity on the stimulated protein, strongly suggesting that it was not working through the catalytic site. So some medchem was done to optimize that from a 5 micromolar inhibitor to a what we'd call sort of a tool compound, which was 71 nanomolar in the in vitro assay, and that inhibitor SHP-099 became the compound that was published in our Nature paper. So to try to understand what the mechanism by which this compound acted as a non-catalytic inhibitor of SHP2, we solved the crystal structure of full-length SHP2 bound to the inhibitor, and I'll show you that now. The 2 SH2 domains are shown on the top in blue and in green, and the catalytic domain is shown here in brown, with the active site highlighted by this red area here. So in the APO form bound to the compound, the 2 SH2 domains form this inactive conformation, shown here with the compound wedged into a hole between the 2 SH2 domains and the catalytic domain. And in this conformation, a loop from the N-terminal SH2 domain but impedes the catalytic pocket. So the catalytic pocket is physically blocked, and the compound itself is found in this pocket, which was previously unknown, between the 2 SH2 domains and the catalytic domain, and actually makes hydrogen bonding interactions with all 3 domains. So our proposed mechanism of action is that this compound stabilizes the inactive conformation of SHP2 and would presumably create a barrier to RTK-dependent activation. I'm not going to show you all the preclinical data that's published in the paper. I'll just show you the in vivo proof of concept, which was to take an EGFR-dependent cell line and treat it with either SHP-099 or erlotinib, an EGFR inhibitor, and you can say inhibiting SHP2 or inhibiting EGFR was equally efficacious in this study. Here's an example of a FLT3-driven leukemia cell line where we got complete regression with SHP-099. Again, it's a fairly early tool compound, so nowhere near the potency that ultimately was achieved for the clinical candidate. So, that illustrates, I think, the importance of thinking carefully about the mechanism of inhibition that you're going for, and especially when there are catalytic domains, thinking more about how to engage the protein by multiple different mechanisms, not just thinking, okay, I have a pocket, let's go for the one we know. And that's going to be further illustrated in the next section, where I'm going to illustrate the development of an allosteric inhibitor of BCR-ABL as a way to try to get around therapeutic resistance to first and second generation ABL inhibitors. And in particular, with respect to BCR-ABL, we were interested in whether or not we could develop a strategy that might lead to a combination regimen that would have curative potential. So I think most people know the story of CML. It's a relatively slow-growing leukemia, which ultimately transforms into an acute blast phase and kills patients. It was the first cancer to be definitively associated with a genetic alteration, namely the translocation between chromosomes 9 and 22, leading to the fusion of the BCR gene and the ABL kinase, leading to constitutive activation of the Abelson kinase. Ultimately, Novartis and then others developed inhibitors of Abl kinase that had substantial clinical activity in this disease. Now, over time, we learned a few lessons about first-generation inhibitors. The first was that mutations can occur in the binding site of the drug that cause resistance to BCR-ABL inhibitors. And when this first was observed, I think people who lived through this era remember there was a lot of hand-wringing about how smart cancer cells were to have developed these resistance mutations, and that this meant it was sort of an intractable problem, and no matter how smart your drug was, cancer cells were smarter. Of course, they're not smart, they're just very good at mutations. The positive side of this was that if you compared the work of Charles Sawyers, which is shown on the left, mapping mutations from clinical samples, and the work of George Daly, where he discovered mutations in an unbiased mutagenesis screen in vitro, there was a strong correlation between the in vitro description of mutations causing resistance and what was described clinically. And at the time, to me, this was very important, because if you thought about all the studies that had been done on Taxol resistance or cisplatin resistance, very little had ever translated between sort of the in vitro study of resistance and the clinical discovery of resistance. In this case, there was a very strong correlation, leading one to think that if you understood resistance preclinically, it would be tractable in the clinical setting. So we were interested in the idea that since most cells were developing resistance through BCR-ABL, that BCR-ABL remained the critical target in CML. I think it's also important to remember that you could have imagined, instead of resistance in BCR-ABL, there could have been 300 pathways that might have been activated to rescue the cell from that one inhibitor. Instead, over and over again, cells chose to mutate BCR-ABL, suggesting this was a critical node for survival of the cells. Now, it turns out that the catalytic domain is not the only domain that has a pocket. In fact, there's a myristyl pocket in the C-terminal lobe of the kinase that helps to create an inactive conformation of wild-type ABL by bringing an SH2 and an SH3 domain into an inactive conformation. I'm not going to go through the drug discovery process that got us to the drug. Initially discovered at GNF was a tool compound called GNF2, which occupied this myristyl pocket. Ultimately, a collaboration between our group in Basel and our group in Cambridge led to the discovery of ABL001, which binds to this myristyl pocket and induces the same inactive conformation as the myristyl binding site. So this is the structure now of ABL001 bound to BCR-ABL, and you can see the critical interactions that are induced by the myristyl group are recapitulated by the small molecule. So the question we asked next was, whether or not, well, first, we were interested in the notion that having targeted a relatively unique site on BCR-ABL, the selectivity of this inhibitor might be quite unique, given that most inhibitors target the catalytic domain, where there's a lot of conservation with other kinases. Having a relatively unique pocket might lead to a very selective profile. And indeed, if you look in the profiles in the Cell Line Encyclopedia, this is the profile of ABL001. It's a single-digit nanomolar cellular inhibitor of BCR-ABL positive cells. It spares all other cell lines in the Cell Line Encyclopedia. It's equally potent to second-generation inhibitors like dasatinib or ponatinib, but you can see has much greater specificity. So it's a very clean inhibitor, and looks like it has activity in the encyclopedia only against BCR-ABL mutant cells. So the question we want to know is, if you have a catalytic inhibitor and you have an allosteric inhibitor, do they have similar mechanisms of resistance or distinct mechanisms of resistance? And you can imagine if they have completely non-overlapping mechanisms of resistance and you were to put them together as a combination, a cancer cell would have to evolve simultaneously 2 mutations in the same cell for one cell to be resistant to both inhibitors. So mathematically, that should be much more difficult than finding one mutation for one drug. So we repeated the in vitro mutagenesis screens that have been done in the past to find the resistance mutations. And you can see this is now using nilotinib, which is a second-generation inhibitor. It creates resistance around the catalytic site, but ABL001 retains activity against most of those mutations except for one, this 359V mutation. When we select for resistance to ABL001, you can see 3 mutations in the allosteric site create a 10,000-fold shift in the IC50 to the allosteric inhibitor, but those 3 mutations are all sensitive to the catalytic inhibitor. So this suggests a largely non-overlapping pattern of resistance. To further test that at the cellular level, we used something called genetic barcoding to track the clonal evolution of cells during the resistance process. So the way this works is we generated a library of random DNA barcodes that were 30 base pairs in length with a forward primer and a reverse primer. No cDNA, no expression, just a tag to introduce into cells. If you make a random barcode of this size, the predicted library size was somewhere between 20 and 80 million unique barcodes, meaning if we infect that library into a cell population, we should be able to track the evolution of around 20 to 80 million individual cells during treatment with an inhibitor. So we imagined that this barcode experiment would work in the following way. If we took KCL22 cells, which have the BCR-ABL translocation, we introduced the barcode library by lentiviral infection, and then apply a drug, and then do next-gen sequencing. If there's a cell that becomes resistant, it will expand, and the barcode will become more prevalent over time, and that the number of barcodes would be indicative of a cell becoming resistant to our drug. Now, you can imagine if instead of just doing that in one replicate, we instead split the cells into 4 replicates and then treated with the drug, if the same barcode pops up in the 4 replicates, it would mean that cell actually pre-existed in the population and was not a de novo acquired mutation. So we thought if we did this in replicates, we could distinguish the idea of spontaneous mutation during resistance from pre-existing mutation in the cell population. If instead of doing the replicates with one drug, we did the replicates with 4 different drugs, we'd be able to ask, were the mutations, the cells resistant to one drug also resistant to 3 other drugs or 2 other drugs or 1 other drug, because each cell would have a unique barcode. So here's what the experiment actually looks like in data. The cells were done with 4 different drugs with 8 replicates for each drug. So there's a lot of cell culture. If you look at the 2 catalytic inhibitors, imatinib and nilotinib, for the imatinib setting, we found 39 unique barcodes that were found in replicates that were resistant to imatinib and grew out over time, meaning the vast majority of cells that became resistant were in the population before we administered the drug. That frequency of pre-existing cells was 0.001% of the starting population. This is well below the detection by NGS. So you could sequence all you want by NGS, you will not see these cells. Similarly with nilotinib, which is a structurally related inhibitor, 32 barcodes pre-existed, also 0.001% of the population. In the case of 2 different allosteric inhibitors, GNF2, which was the predecessor tool compound, and ABL001, 150 or 160 barcodes pre-existed in the population that were resistant, representing 0.006%. So one lesson here was, actually there are more cells that are resistant in the starting population to the allosteric inhibitor than to the catalytic inhibitor. And in fact, we expected this because the allosteric site is an inhibitory site. So there should be mutations that can activate the kinase even a little bit through the allosteric site, and that should be easier than getting a mutation in the catalytic site. So the question is now, are the cells that are becoming resistant in these barcoded the same or different? So we can just align those barcodes and look at whether they are or aren't different. You can see there's more shared resistance between the two catalytic inhibitors and between the two allosteric inhibitors. And in fact, there was only 3 shared cell barcodes that were resistant to the allosteric inhibitor and to the 2 catalytic inhibitors. It turns out these were the gatekeeper mutation, T315I, which was not inhibited by GNF2, but is inhibited by ABL001. So between ABL001 and either imatinib and nilotinib, we saw no shared resistance. So no barcode emerged in those populations that was identical. So that suggested that a combination of ABL001 with either imatinib or nilotinib might prevent the emergence of therapeutic resistance. And so to test that, we took those same cell lines and we did an in vivo experiment where we implanted the cells, established the tumors, and then treated them first with nilotinib. You get complete remission, and then they grow out, and they grow out with the gatekeeper mutation shown here. If you do the experiment the other way, if you then take those cells and you switch to the allosteric inhibitor, you regress those tumors, but then you get resistance again to the single agent. If you do it in the reverse, you treat with the allosteric inhibitor, you get complete regression, but then you detect a mutation in the allosteric binding site. If you now switch to nilotinib, you get regression again and resistance. So I'll only point out this is the vast majority of clinical trials that are done today. Treat with one drug, wait till the patient gets resistant, and now switch to the other drug. So the idea that that's going to work as a therapeutic strategy, I think should, we should have great pause about that. If instead of doing the sequential therapy, we treat from the beginning with the 2-drug combination of nilotinib and ABL001, we never saw therapeutic resistance emerge, and we stopped dosing at day 80 and no tumors regrew out to day 180. So in the mouse experiment, with whatever volume of cells we implanted, we're able to, it looks like, eradicate the tumor. So a few points about that. One is most people that and this has included my groups at Novartis, that were studying combinations use synergy as some important determinant of whether a combination should be a good combination or a bad combination. These 2 drugs have no synergy because they're targeting the same protein. It's very hard to have 2 active drugs that are both active that show synergy. They're barely additive in your traditional synergy experiments. In fact, what's very interesting is there's some synergy with imatinib because imatinib is a weaker inhibitor. There's almost no synergy with nilotinib because nilotinib is a more potent inhibitor, and it's very difficult to show any synergy when you already have a potent inhibitor. Yet, this combination showing no synergy eradicates the tumor because it has non-overlapping cross-resistance. So my own opinion is if I were to develop a combination, I want 2 drugs that are both highly active against the tumor and show non-overlapping cross-resistance, not necessarily synergy. So this combination is in the clinic. I only have, since I left Novartis, at the time I left, we only had the phase 1 data for ABL001, so I can only show you what's been publicly disclosed. We've shown 1 patient in detail where we really feel like we've shown that at least the allosteric inhibitor is working by the intended mechanism. This was a patient that came in that was on dasatinib. You can see had a 1 log reduction in its transcript, and then the patient relapsed with nearly blast crisis. So this patient was almost a log higher in transcript numbers when the patient came in to ABL001. You can see the patient had dramatic response, even better than dasatinib, to the single agent of ABL001, but then relapsed, and actually relapsed with 2 different allosteric site mutations. One that we had previously seen, which is this V468F mutation, which we previously saw, And the second mutation, I502L, which is actually also right near the binding pocket, but didn't show up in our preclinical screens. So this suggests to us that ABL001 has single agent activity through the allosteric binding site. It's proof of mechanism that the drug is working by its intended route of activity. And hopefully now when the combination trials start, we'll hopefully not see the emergence of therapeutic resistance. So to summarize, I think I really feel like understanding that disease pathogenesis has enabled us to develop transformative medicines. The large-scale functional genomic screens that I described unveiled this synthetic lethal interaction between the deletion of MTAP and the dependence on PRMT5. The first therapeutic inhibitors that will take on that synthetic lethality are now in the clinic from Agios Therapeutics. I've shown you that creative biochemical screens and thinking a little harder about the mechanism by which proteins were activated and inactivated led us to the discovery of novel allosteric inhibitors of SHP2, which are now in clinical trials. And then the dual targeting of BCR-ABL through this combination of an allosteric site inhibitor and a catalytic inhibitor eliminated resistance preclinically, and I have great hopes that it would do so clinically as well. So as Mark was saying, I'm back in academia now, and so I'm still recruiting postdoctoral fellows, so if people are interested, that's my email. Hopefully you won't transmit that email to telemarketers, but I'd be happy to hear from people who are interested in what we're doing. So thanks very much for your attention.
Take some questions. Tima, right there behind.
Hi, really great talk. I have a question about your cell lines that you've done the polar metabolite profiling of. Are you guys also looking at lipidomics too?
So lipidomics was not done in that, though. Some lipids are captured, but specific lipidomics, no. So, but there are lipids in those profiles. So like the acyl carnitines are in there as well.
And I had one other question. So you said that there was no strong metabolomic profile for the single gene mutations like P10. That was across all the cell types. Do you see specific profiles when you break it down between like pancreas or colon or?
I think the problem with that is you become, even though it's a large collection, you become very quickly limited by the statistical power to do that. So each time you break it down into a tissue type, you're ending up with 3 or 4 P10 null cells, for example. So I think probably only in the P53 setting would you have the power to do that.
Thank you.
More questions?
I wanted to ask a general question, actually 2 questions. So one, you made the comment about the approaches for doing trials, and a lot of times we hear in the clinical setting that it's very difficult to do a combination trial because of the companies not wanting to interact. Could you make a comment about that?
Yeah, I think that is difficult. For 2 companies to take, there's 2 reasons there's difficulty. One is, let's say when I was at Novartis, our strategy was to actually develop the combinations. So if we had our own BRAF inhibitor or our own MEK inhibitor, but someone wanted to take our MEK inhibitor and combine it with someone else's BRAF inhibitor, yeah, we weren't so interested in that. I think the other concerns are how the interaction from a safety perspective plays out and how that then influences the other development pathways that a set of inhibitors might be undertaking. So I think those are challenging. It's not to say that they haven't been done, though. So there are more and more examples of those things happening. It's certainly much, much easier once the drug's on the market than when it's in the middle of development.
The other question related to that was, it's very impressive what you've shown, going from either identification of a driver through whether it's genetic or through some other screen. And in looking at a lot of academic data. I'm just wondering if you have any suggestions as to, and whether it's in the setting now precision medicine, how in academia we can, really help get to the point where we're doing things where you're actually going to whether it's a tool compound or getting to a drug, because really we're seeing a lot of identification of drivers potentially, but things just don't move forward beyond that in academia. I'm just wondering if you have any thoughts.
So do you mean on the actual therapeutic side?
Yeah.
I think so first of all, it takes way more investment than people realize. And I'd say the second is, what I've seen happen over the last decade is that in academia, people have equated drug discovery with screening, or HTS. And HTS is unbelievably noisy. But yet I see papers where someone does an HTS and they pull one compound out and start working on it. Well, I have no idea how you can do that. I'll give you another example of what it's like to do an HTS. We ran an HTS, we've presented this publicly, on MCL1. So MCL1 is a protein-protein interaction. It's a difficult target. We screened 1.8 million compounds, 12,000 hits. 12,000 hits were taken through a series of flowcharts, reproducing the IC50, differential scanning calorimetry, mass spec to rule out covalent binding. Obviously, we also had a off-target profile, NMR characterization, and then structural characterization. So from the 12,000 hits, we had 6 validated hits. So that's the false positive rate. The false positive rate was 11,994 false positives and 6 true positives. So people conflate HTS with drug discovery. HTS is hit finding. And it's not just running the screen. If you're really going to do that, you need to have a set of validated assays to take all of the hits through and find out what you really have. If I were to invest in drug discovery, I wouldn't build an HTS screen. I would think about things like structural biology and biochemistry and understanding protein interaction and protein mechanism as much as possible at the genetic level till I had precise insight as to what type of therapeutic I wanted. And the more precision you can get from a genetic point of view, the more precise you could be in exactly how to tailor a thinking around a therapeutic, rather than let's take MIC and screen. Yeah, good luck.
On that note, to invite everybody for an apéro outside. And Bill, thanks so much for your incredible lecture.
Thank you.